This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. In conventional breeding, breeders manipulate the whole plant genome at once. When they want to insert a new trait in an existing plant genotype, they have to cross at least 2 different varieties to obtain an F1 hybrid. If the breeder further wishes to obtain a pure line, they need to proceed with several generations of controlled crosses, either selfing or back crosses, in order to produce a purely homozygote line that has all the desired traits. This requires at least 6 to 10 years of selection for annual crops and much longer for perennial ones. Moreover, the efficiency of conventional breeding is limited by the reproductive compatibility of organisms. Crossing between different plant species is complicated because of the existence of reproductive barriers. These barriers can be removed under some circumstances. But what if a breeder would like to improve the plant so that they express a bacterial trait? How do we even go about crossing a plant and a bacterium? The answer to this is genetic engineering. Genetic engineering is simply defined as the precise modification of an organism's DNA by inserting DNA from another organism. Depending on the source of the inserted DNA, we distinguish between cisgenesis, where a gene from the same species is inserted in a target organism, transgenesis, where a gene from another species is inserted in the target organism, and more recently, subgenesis, where no foreign DNA is inserted, but the plant is induced to make its own targeted genome modifications. Classical genetic engineering follows several steps. First, a gene construct is prepared. This construct is a synthetic DNA sequence that will be inserted in the modified organism. It contains at least 4 elements: a promoter sequence, which initiates and regulates DNA transcription, a gene of interest, which codes for one or multiple traits that the breeder wishes to improve in the plant, a reporter gene, which indicates whether the insertion of the gene construct was successful, and a terminator sequence, which indicates that the transcription of the construct should be stopped. Once the gene construct is ready, it needs to be inserted in the cell of the crop that we wish to transform. This can be done using several methods, such as mechanical insertion or transformation with agrobacterium. After the insertion, the plants that have been successfully transformed are identified by screening. The reporter gene is very important in this step, as it helps to quickly identify plants that express it, because they show a phenotypic trait that is easy to recognize, such as fluorescence. But once the genetic transformation is successful, the reporter gene needs to be removed, so that the engineered plants only have the traits of agronomic interest and no traits related to the reporter gene. These steps are less or more the same for all classical techniques of genetic engineering. Subgenesis, also known as gene editing, does not require the use of a gene construct. There are different technologies used for gene editing, but all of them are characterized by 2 components. The first one recognizes and binds to a specific DNA sequence in the genome that we wish to modify, and the second one is an endonuclease which cleaves the DNA at a specific recognition place. In the following videos of this playlist, you will see how each of these techniques works in details and examples of their application.